Hi, my name is Anika Chakrabarti. I'm a junior at Lexington High School this year, and I'll be talking about epigenetics and neurological disorders. So the research question that I was trying to answer is how the dysregulation of epigenetic mechanisms are linked to neurological disorders such as Huntington's disease and Alzheimer's disease. Epigenetics has a lot of diverse mechanisms such as DNA methylation, histone and non-coding RNAs, and they all hold significant therapeutic potential for a lot of different disorders and diseases. So I thought that epigenetics could be leveraged for a bunch of tailored medical approaches that targeting specific and extensively researched conditions. Epigenetic machinery are essential for our molecular and cellular processes to function, but there are various factors that can disrupt these mechanisms. So if there's a correlation or causation between the dysregulation of epigenetic mechanisms and neurological diseases, then it could help scientists discover therapeutic strategies for Alzheimer's and Huntington's disease, as epigenetics is already a really well understood and well studied biological system. So to first understand epigenetics, we first must need to understand the structure and organization of genetic information. So DNA on the leftmost diagram, which is number 5, is wrapped around these proteins called histone proteins. And those histone proteins are then called nucleosomes, number 4. That is then strung together to form the chromatin fiber. You can see that on number 3. That chromatin fiber is then compacted together to create condensed chromatin, which is then formed and compacted even further into the chromosome shape that we're used to seeing, which is represented by number 1. Chromatin is one of the most important structures when discussing epigenetics. Chromatin is found in two distinct forms, you can see on the right diagram, heterochromatin and euchromatin. When chromatin is condensed, it is called heterochromatin. In heterochromatin, gene expression is repressed, and when chromatin is not condensed in euchromatin, that is the state in which gene expression occurs. So what is epigenetics? Epigenetics regulates gene expression without actually altering the DNA sequence itself. So it basically does this by changing the form of chromatin into either euchromatin or heterochromatin that I was mentioning earlier. It also studies how individual genes and gene networks function at different molecular and cellular levels, and it aims to understand the mechanisms by which gene expression and function are controlled. So I'll be talking about first various epigenetic mechanisms, and I'll start with DNA methylation, which is the most commonly known and talked about. So this is when methyl tags are added to the chromatin, and this causes chromatin to condense. So genes that are targeted for transcription are inaccessible and therefore unable to be transcribed. Or in other words, genes are repressed. DNMTs are the primary enzymes that facilitate this process. Histone acetylation, on the other hand, is essentially the opposite process. So histone proteins are tagged with acetyl groups, and that causes the chromatin to relax and therefore expand. This form lets genes be accessible for transcription, and so target genes can be expressed. Non-coding RNAs can either increase or decrease expression. They don't code for proteins, but they're involved in numerous processes. ncRNAs work by binding to other RNAs to block their expression or for degradation, which means destruction. On the other hand, it can also enhance gene expression by modifying the structure of chromosomes in a way that makes the genes easily accessible to transcribe. An example that I wrote about was microRNAs, which inhibit stability or repress translation of target RNAs meant to become proteins. RNA editing, on the other hand, is a mechanism where protein coding genes can be modified and regulated by RNA transcripts. And thus, this process adjusts the expression, profiles, and functions of different protein coding genes and non-coding RNAs. So now let's talk about the dysregulation of epigenetic mechanisms. So DNA methylation inhibitors sequester or hide away DNMT enzymes, which we talked about earlier that catalyze the process of methylation. So this actually reactivates genes that should be silenced. We can see this through a test done where these inhibitors were administered into the brain tissues of mice and rats, and scientists found that these enzymes, the DNMT enzymes, were unable to methylate chromatin. And so this led to an unintentional and sometimes overexpressed state of certain genes. Also, lifestyle factors influence epigenetic patterns significantly, and that includes factors like diet, obesity, and exposure to alcohol and tobacco. As you can see in the diagram I made here, it differentiates which factors modify which mechanisms. But a lot of these studies are done on the effect of these factors on DNA methylation in particular, because DNA methylation is the most talked about and studied part of epigenetics. So now I'm going to talk about the correlation between the dysregulation of these epigenetic mechanisms and neurological diseases. So what is Huntington's? Huntington's disease is a genetic disorder that primarily affects the neurons. It's caused by genetic change in the gene HTT, or the Huntington gene. And this gene codes for a protein that is essential for the proper functioning of neurons. So how does this relate to epigenetics? Well, there has been research showing that in the brains of HD patients, there are irregularities in the gene transcription levels and patterns. In addition, in genes that displayed altered expression from mutant HTT, there were also found to be observable and significant changes in the DNA methylation of that gene sequence. But it's important to note that this research investigating the correlation between dysfunctional epigenetic mechanisms and the diseases indicates that these mechanisms are more of a downstream effect rather than the root cause of the mutation of the HTT gene. Alzheimer's disease is the most common form of dementia and affects the elderly population. It's categorized by the ongoing and progressive neurodegeneration in specific regions of the brain. This disease is associated with the accumulation of the amyloid B peptide in the brain, or in other words, protein aggregation. So too much of this protein in areas where it shouldn't be. Dysregulated epigenetic mechanisms in Alzheimer's disease include DNA hypermethylation, deacylation of histones, and a general repressed chromatin state. In AD, the methylation levels of the amyloid precursor protein, or APP, promoter region on the gene in the temporal lobe are significantly lower compared to those in healthy individuals. This lower amount of methylation results in higher expression, leading to increased protein production and worsening of the condition due to protein aggregation, mostly of that amyloid B peptide protein in the brain. So studies have shown that the AD brains exhibit a large number of differentially methylated or hydromethylated regions compared with control brains. And then this suggests that the dysregulation of epigenetic mechanisms can potentially serve as a biomarker for downstream correlated factors in Alzheimer's disease. But just like in Huntington's disease, it's important to note that the role of epigenetics in AD is considered as downstream and correlated to AD rather than being a cause. So epigenetics' intrinsic adaptability prevents DNA from enduring permanent damage, while at the same time regulating gene expression in reversible and plastic ways. Epigenetics could be leveraged for tailored medical approaches that target specific and extensively researched conditions like Alzheimer's and Huntington's. Each individual possesses a distinct epigenetic profile that's different and that can change for their specific needs and requirements, which is why scientists often call it plastic because it can change. However, there are lingering questions pertaining mostly to the efficacy of this intervention in epigenetics because there are a lot of risks that can be associated with interfering in this intricate system, given its inherent connection to the other molecular and cellular processes that coexist with epigenetics. Thank you.
Wonderful, wonderful, Anika. And sorry I called you Anika. It's Anika, everyone. Anika, fantastic work. We did have a couple questions for you. So the first one is, can environmental factors like stress or diet trigger these epigenetic changes?
Yeah, they can, especially stress often leads to diet changes, and diet is one of the biggest environmental factors that can especially affect methylation.
Awesome. The other question is, are there treatments being developed that target epigenetic problems in the brain?
Currently, I don't think there are treatments that are being developed, but I think the first step is researching ways that epigenetics can target these conditions in the brain before therapies can be developed. So I think right now there needs to be more research done on it.
Awesome. And what advice would you give to another student that's just starting out with this research topic?
I think I would advise them to focus in on especially what they're most curious about. If you're most curious about like neurological disorders, then I would focus on that instead of going too broad, because that can be a little bit disorienting.
Awesome. Any other questions? See if any other came in. Okay. I think that's all. Wonderful job, Annika. Congratulations on finishing your
Yeah,
presentation.
thank you.
No worries.